Usually when we talk about the brain, there's this expectation of it being this isolated fortress.
Right, like an impenetrable vault.
Exactly. We picture the blood-brain barrier acting as this, ultimate security gate. The idea is that it keeps all the systemic chaos of the rest of the body out.
Yeah, allowing the brain to just quietly do its highly complex, pristine work.
Right. It's a very comforting view of human biology. But, well, then you step into the world of neurodegenerative disease.
Specifically Alzheimer's.
Yeah, and suddenly that fortress wall looks, a lot more porous. Actually, based on the stack of sources we're doing a deep dive into today, the fortress metaphor just entirely collapses.
It really does.
We are looking at a groundbreaking 2026 study. It was published in the journal Cell by Huang and colleagues. And the mission of today's deep dive is to explore this wild, newly discovered mechanism behind Alzheimer's disease.
And it is wild. We're talking about cancer-associated genetic mutations in the immune system secretly driving brain degeneration.
It completely flips our standard understanding upside down, right? Like, imagine if the immune cells meant to protect your brain went rogue, started accumulating mutations like a tiny tumor, and just began setting fires instead of putting them out.
The paradigm shift here really cannot be overstated. We are so conditioned to associate Alzheimer's solely with amyloid plaques and tau tangles.
Right, the classic protein aggregates building up in the brain tissue.
Exactly. But this paper proves that somatic variants, these genetic typos acquired over our lifetime, are playing a massive, previously hidden role. We aren't just looking at a brain struggling to clear waste anymore.
No, we're looking at an active infiltration by rogue, mutating immune cells. So let's get into the mechanics of that, because the biology here is just fascinating.
To do it.
So we're assuming you, the listener, are familiar with the basics of DNA and genetics. You know that we inherit germline mutations from our parents.
Right, the misprints from the factory, so to speak.
Right, but this study focuses entirely on somatic variants, specifically things called SSNVs and SCNVs, or MCAs.
Yeah, let's unpack those acronyms so we don't leave any details behind.
Please do.
So an SSNV is a somatic single nucleotide variant. It's a typo in just one single letter of your genetic code.
And the other ones?
The SCNVs and MCAs stand for somatic copy number variants and mosaic chromosomal alterations. Basically, instead of just one wrong letter, these are massive structural changes.
Yeah.
Like tearing a whole page out of the DNA manuscript or duplicating a whole chapter.
Okay, I see. So if our DNA is a manuscript, germline variants are those misprints from the printing press, but somatic variants are the typos or ripped pages we make while furiously rewriting the manuscript throughout our lives.
That is a perfect analogy, and we acquire them constantly. Every time a cell divides, there is a tiny probability of a copying error.
Right.
Most of these somatic variants are completely benign. But occasionally, a spontaneous typo hits a critical regulatory gene. And when that specific type of mutation happens in a blood stem cell inside your bone marrow, it triggers this cascade known as clonal hematopoiesis, or CHIP.
CHP. Okay, let's break down why clonal hematopoiesis is so dangerous. A stem cell in the bone marrow is basically, a factory worker, right? Constantly pumping out red and white blood cells.
Right, billions of them.
So if that stem cell acquires a mutation that breaks its internal regulatory brakes, it doesn't just keep functioning normally, it gains this distinct survival advantage over the healthy stem cells around it.
Exactly. You basically have this microscopic Darwinian evolutionary battle happening right inside your bone marrow.
Wow.
Yeah, that one mutated stem cell starts dividing slightly faster, and its offspring live slightly longer than their healthy neighbors. Before long, that single rogue cell has cloned itself endlessly.
Hence the clonal in clonal hematopoiesis.
You got it. And suddenly, a significant percentage of your circulating white blood cells are carrying this specific acquired mutation.
Which brings us to the brain's gardeners, the microglia. They're the cells that usually clean up debris in the central nervous system. Okay, so let's do a quick recap for you, the listener, before we move on. Number 1, somatic variants are acquired DNA typos. Number 2, some of these typos give blood stem cells a survival advantage, causing runaway cloning. And number 3, our brain has its own immune gardeners called microglia.
Spot on.
So here's a question for you to ponder. Have you ever considered that your immune cells are constantly evolving and competing with each other right now inside your own bones?
It's a bit unsettling when you really think about it.
It really is, but it raises an obvious question for this study. If millions of people are walking around with these mutated, hyper-competitive blood cells and these mutations are driving Alzheimer's, why did it take until 2026 to find them in the brain?
That is the million-dollar question. And it comes down to the physical limits of older sequencing technology.
Like whole exome sequencing.
Exactly. Previous genomic studies relied heavily on whole exome sequencing, or WES. In standard practice, they were sequencing at about a 70x depth.
Meaning they read each letter of DNA roughly 70 times to ensure it was accurate.
Yes. The problem is that depth can only reliably spot a mutation if it is present in, say, more than 5% of the cells in a given sample. This is called a high variant allele fraction, or VAF.
Ah, okay. So if these mutated cells only made up 2% of the brain tissue sample, the old sequencing algorithms just threw the data out?
Basically, yeah. They discarded it as background noise. The algorithms couldn't distinguish a rare biological mutation from just a standard error made by the sequencing machine itself.
Right.
To bypass this, Huang's team used deep panel sequencing. They didn't read the DNA 70 times, they read it over 1,000 times.
Wow, 1,000 times. But depth alone isn't enough to solve the background noise problem, is it?
No, it's not. They also had to use UMI barcoding, which stands for unique molecular identifiers.
I love the mechanism behind UMIs. It solves such a massive physical problem. Like, think of it this way. If you take a single photo of a complex puzzle piece and see a scratch, you might wonder, is the scratch actually on the puzzle piece, or is it just a smudge on my camera lens?
Right, the base calling errors in sequencing are the smudges on the lens.
Exactly. But if you attach a unique chemical barcode, a UMI, to the original DNA molecule before you amplify and copy it, it's like taking 1,000 photos of that exact same puzzle piece.
That's a great way to visualize it.
If the scratch shows up in every single photo that carries that specific barcode, you know it's not a camera smudge, it's a real physical scratch on the molecule.
Highly accurate. By combining UMIs with the 1,000x depth, the researchers could confidently identify mutations present in as few as 1% of the cells.
That's incredibly precise. And they applied this to the ROSMAT cohort, right? Like 190 Alzheimer's brains and 121 healthy controls.
Yes, a massive sample size. And the findings were stark. The Alzheimer's brains had significantly more mutations in a panel of 149 known cancer driver genes.
And specifically in tumor suppressor genes, the TSGs?
Right. We usually think of tumor suppressors as the brakes on a car. If you cut the brake lines, the cell just accelerates and multiplies indefinitely.
And the researchers found that the specific brake lines being cut were primarily in 3 genes, TT2, ASXL1, and DNMT3A.
Yes, those were the top mutated genes.
So we're going to give you a mnemonic to remember these. Think of the word TAD, TAD, TET, ASXL1, DNMT3A. These are the bad actors. The TAD genes.
I like that, the TAD genes. And to prove these CAD mutations were actually driving the disease, the researchers used a mathematical test called the DNDS ratio.
Okay, break that down for us.
So the DNDS ratio compares non-synonymous mutations to synonymous ones.
Wait, meaning what? Exactly.
A non-synonymous mutation is a typo that actually changes the physical structure and function of a protein. A synonymous mutation is a silent typo. It changes a letter, but the resulting protein is exactly the same.
Okay, so if mutations were just accumulating randomly as we age, the ratio of functional changes to silent changes should be pretty balanced.
Exactly. But that's not what they found. They found a heavily skewed high DNDS ratio for the TAD genes.
Meaning positive selection.
Yes. It's the mathematical fingerprint of positive selection. It proves these mutant cells aren't just passively sitting in the brain, they are actively outcompeting healthy cells.
Okay, time for another quick 5-minute recap for our listeners. Number 1, old sequencing missed these mutations, but deep sequencing with UMIs caught them. Number 2, Alzheimer's brains are full of typos in tumor suppressor genes, specifically the TAD genes. And number 3, these mutations cause runaway cellular cloning.
Perfect summary.
But this brings us to a huge contradiction. Like, if these are blood mutations born in the bone marrow, what are they doing inside the brain?
That is the sleeper agent twist.
Right. So to figure out where the mutations were hiding, they had to sort the brain cells.
Yes, using FANS, fluorescence-activated nuclei sorting. They essentially broke the brain tissue down into individual cell nuclei and tagged them with fluorescent markers.
And they tagged microglia with CSF1R and neurons with NUN.
Right, and what they found was that the TAB mutations were almost entirely absent in the neurons. They were heavily concentrated in the CSF1R positive cells, the microglia.
But here's where it gets really interesting. The exact same mutations were found in the patient's paired blood samples.
Yes. The DNA from the circulating blood matched the mutations in the brain tissue perfectly.
Wait, hold on. If these are blood mutations and we are pulling them out of brain tissue, Does that mean the blood-brain barrier is broken?
It means it's actively failing. As we age, the blood-brain barrier breaks down, and endothelial cells lose their tight junctions.
And the blood cells just sneak in.
Basically. Bone marrow-derived monocytes, which are white blood cells, detect inflammatory signals from the brain and squeeze through those microscopic leaks.
And they don't just stay monocytes, do they? Once they cross into the brain, they disguise themselves. The paper calls them MLBMs, or microglia-like brain macrophages.
Exactly. They put on the uniform of the local security force. They look and act like the brain's resident microglia, but they are secretly mutated invaders from the blood.
So, to summarize for you listening, the brain isn't just decaying, it's being infiltrated by mutant sleeper agents from the blood. Which leads to this question for you. What if Alzheimer's isn't just a brain disease, but fundamentally a blood and immune system issue?
It's a completely different way to look at dementia.
Okay, so we've caught the sleeper agents in the brain, but what exactly is their mission? How do these MLBMs cause Alzheimer's once they are inside?
Well, the researchers answered that by looking in vivo in human brains using techniques like GATI-TA and SciType.
GATI-TA, that stands for genotyping of targeted loci with single-cell chromatin accessibility, right?
Yes. They used that alongside the C-AD cohort to map out not just which cells had the mutation, but how the mutation was physically altering the DNA's behavior inside the cell.
And they found these mutant MLBMs shift into a biological state called DAM.
Disease-associated microglia.
Right. Normally, microglia are in this peaceful homeostatic state. They just clean up dead synapses and nurture neurons.
But the DAM state is a wartime scorched earth defense protocol.
Yeah, they become hyperproliferative, they multiply wildly, and they start pumping out neurotoxic inflammatory cytokines, setting the fire, basically.
Exactly. And the reason the TAD mutations cause this comes down to epigenetics.
Right, because TESI2, ASXL1, and DNMT3A are epigenetic regulators.
Yes. Their normal job is to dictate how tightly DNA is coiled and to add or remove methyl groups to turn other genes on or off.
So, like, DNMT3A normally adds methyl groups to silence inflammatory pathways when they aren't needed, right? It acts as the biochemical brakes.
Spot on. When you break DNMT3A with a somatic mutation, the cell loses its ability to regulate its own genome. The DNA unravels in all the wrong places.
So the genes that control the scorched earth DAM state get stuck in the on position.
Right. The cell is physically incapable of returning to its peacetime role.
Okay, I have to push back here, though. Because if we're looking at human brains that have already died of Alzheimer's, we're looking at a crime scene after the fact.
That's true.
So correlation isn't causation. How do we know the toxic amyloid-filled environment of the Alzheimer's brain didn't cause the infiltrating cells to panic and shift into the DAM state?
That is the ultimate hurdle in observational science. To prove causation, they had to recreate the environment from scratch in vitro.
The CRISPR experiment.
Yes. This is where the experimental design is just brilliant. They took healthy, human-induced pluripotent stem cells, or iPSCs, and crucially, they used a cell line with an APOE3 genotype.
Okay, let's highlight that. We know APOE4 is a massive genetic risk factor for Alzheimer's, but APOE3 is the neutral, healthy baseline.
Exactly. They took this neutral blank canvas and used CRISPR-Cas9 to intentionally edit in our TAD mutations.
They purposefully broke the tumor suppressors in a petri dish.
Yes. Then they coaxed those edited stem cells to develop into IMGLs, induced microglia-like cells.
But I mean, a petri dish isn't a human brain. How does an edited cell in a lab mimic decades of aging?
Because of how the cells behaved the moment they matured. Even in a completely sterile dish, with absolutely no amyloid plaques, no tau tangles, the mutant cells autonomously shifted into the toxic DAM state.
Oh, wow. So the genetic typo alone was enough to trigger Alzheimer's-like inflammation.
The typo alone. And they even caught the cells changing their metabolism.
No.
The mutant cells switched to glycolysis.
Glycolysis. Wait, normally immune cells use oxygen to generate energy, right?
Right, it's efficient. But glycolysis is a fast, incredibly dirty way to burn glucose. It's actually the exact same metabolic rewiring we see in solid tumors, known as the Warburg effect.
So they literally rewire their engines to burn dirty fuel just so they can quickly build an army of clones and spit out mass amounts of inflammation.
Exactly, it proves causation. The somatic variant directly flips the switch to neurodegeneration.
Okay, final 5-minute recap for you. Number 1, the mutated cells are MLBMs, blood cells that infiltrated the brain. Number 2, in human brains, these cells get stuck in a toxic inflammatory DAM state. And number 3, stem cell experiments prove that these cancer-like mutations directly cause this toxic state, completely independent of traditional Alzheimer's risks like APOE4.
That is the perfect summary of the data.
Which brings us to the big picture viewpoint here. For decades, we've been hyper-focused on clearing amyloid plaques.
Right. The pharmaceutical industry has spent billions on it. And while those plaques do create a toxic environment, this research exposes a massive blind spot. Because even if you clear the amyloid, you still have this army of clonally expanding mutated blood cells pouring gasoline on the fire.
But this also offers a really exciting, hopeful application, doesn't it?
It really does. Since these mutations disrupt pathways we already know about in cancer, like the PI3K, PKB ACT survival pathway, we don't have to start from scratch.
Because we already have cancer drugs.
Exactly. We have FDA-approved oncology drugs designed to target these exact clonal blood diseases. We have inhibitors that suppress these overactive pathways in leukemia.
So we might literally be able to treat Alzheimer's using existing cancer drugs.
Conceptually, yes. We could potentially clear the road immune cells from the bloodstream before they ever have the chance to breach the blood-brain barrier.
That is just incredible. So what does this all mean for you, the listener? We used to think of the brain as this isolated fortress. But if rogue, mutating blood cells can breach the walls and cause Alzheimer's over decades, it changes everything.
It demands a completely holistic view of human biology.
It really does. So here's a final provocative thought to ponder. If lifetime mutations in the blood can cause Alzheimer's, could other mysterious neurodegenerative diseases like Parkinson's or ALS actually be blood-driven immune disorders in disguise? Next time you get a routine blood test, think about the secret evolutionary battle happening inside your veins right now.
It's a fascinating thought.
It really is. Let us know what you think of this deep dive.